Going from a gene to a protein, it's an overview of what's going on today. And now I'm talking about this exercise from the DNA, talk about DNA before, to mRNA, and now I'm going to talk about how you make the actual protein itself. And that's done through translation. And then we'll talk about how the signal gets to the peptide, gets to a specific place. It needs to be in the molecule, and the role of RNA, and mutations, and overall what a gene is. So, translation is the RNA thread synthesis of the polypeptide, and we see it happens over here in this figure. So basically, you have the messenger RNA over here on a single strand, and it's and that contains the sequence that's contains the code to make protein. So it has three nucleotide bases to make a codon, and that's what's being read to make protein. So that the transfer RNA comes over, finds the correct three-prong code, and then it's going to bring this up an amino acid attached to this code, attached to this ribosome, and this mRNA sequence, and attach the amino acid and make a polypeptide. So it's basically a cycle of initiation, elongation, and termination. This keeps going on and making more polypeptides. And on one end of the tRNA, so you have a complementary codon right here. This is called anticodon. So mRNA you have a codon, and on tRNA on the bottom half you have an anticodon, which is an important aspect, and that binds to the mRNA. On the other side of tRNA you have the amino acid. So that's basically the most important part of tRNA. And then it brings the amino acids just floating around in the cytoplasm. tRNA will grab onto the amino acid and then bring it to the mRNA. Bring it to the ribosome and make a polypeptide.
You got a question?
Yes.
How do the amino acids attach to the tRNA? You see how it's like different amino acids being connected to one tRNA?
Actually, that's brings me to my next two slides. There's a tRNA synthase that actually matches the correct amino acid to the tRNA.
But since each tRNA codes for a different amino acid, how are they linking different? Like if you look at that picture, you're seeing different amino acids being linked to one tRNA, and how does that happen? And it only has one anticodon.
Oh, well, actually, so it's actually a process. This is more than one tRNA right here. So this peptide essentially will stay the same, and then this tRNA will bring another peptide and add to it. So that's what's happening. I'll get to more specifics of that in the next picture. Okay. So quick question: where's tRNA made? What do you guys think tRNA is made of? Anyone? Well, it's a ribosome. So it's a protein. So it's made in nucleus, goes to the nucleolus, and comes out to the cytoplasm. So it's just like another protein that has a function. Translation overview, structure and function. So tRNA is really important. It goes to the cytoplasm, picks an amino acid, and it deposits to the ribosome. It corresponds to the codon. So here's the structure of tRNA. Here's the anticodon here, and it has the complementary codon to the mRNA on the bottom half. So the top half is the amino acid attached to the site. And it's actually one strand. It's actually a one-stranded molecule. And what happens is the hydrogen bonds between the bases make it into a 3D structure. So that actually is by chance that it makes an L-shaped structure, and that it is a functional unit. So like this is an example of like kind of like a ribosome. It's a functional RNA. Okay. And also, so 17.5 or 17.4 in your book, there are 20 amino acids. There are 20 amino acids, and there's only 4 nucleotide bases. So it's the unique combination of those 4-letter codes that's actually able to bring the amino acids to the mRNA. So genetic code is essentially redundant. So you have 64 possible codons. You have 4 bases, and there's 4 cubed, makes 64, so 64 possible codons coding for 20 amino acids. It's essentially the genetic code is redundant. It repeats itself. As possible because the third base is actually not as strictly enforced as far as attachment goes. third base is called wobble base. Any questions so far? Real quick. Third base is called wobble base. And real quick on the bases. So you have the codon. Third base of the codon, you get U, C, A, or G. And you get U, C, A, or G, and anticodon G, A, U, or C. And you know U attaches to G, U attaches to A, and A attaches to U, G, and C. So it's also that A and U is kind of unique, two unique nucleotides in importance. So, each specific amino acid is captured in the amino cell tRNA synthetase. It's a hard word to say. And there's 20 different synthases for the 20 different amino acids. Each one has their own unique synthetase. That's really hard to say. So what happens? The amino acid will bind to the synthetase, and then the GTP will bind to it. And then it's phosphorus, and then it's and then the tRNA will come, and the GTP gets phosphorus, it gets a cleaved off the GTP, and that basically is the coupling reaction that allows the covalent attachment of the polypeptide to the tRNA. It's over here. So there's a covalent attachment powered by what calls the GDP, energy coupling. And it's up to synthetase is really important because that's the marker that's going to ensure that the correct amino acid gets to the mRNA. Now the functional part of the whole process is the ribosome. It's the most important part. How many subunits are there to the ribosome? 2? Okay. Yes, right. That's right. Large and small subunits. Okay. And where do the subunits come together? Anybody have an idea? So you have 2 subunits, the large and small subunits, and they're actually separated in the cytoplasm. And then the mRNA will bind to the small subunit, and then the mRNA will bind to the small subunit, and then the large subunit will bind after the mRNA, the tRNA binds to that. It's actually a process of them coming together. And ribosomes are actually pretty similar to eukaryotes and prokaryotes. And basically, like evolutionarily, we're more complicated as humans than we are as bacteria. So we have 4 subunits versus 3 subunits. And actually, for your health questions, students, antibiotics can actually target the ribosomes and alter and stop and halt translation based on size. So if you see tetracycline, what happens is you have these, you can identify the fact that a prokaryote has 3 subunits to this 3 RNA molecules in this ribosome, and that a eukaryote has 4 RNA molecules in the ribosome. And because of that difference, it can actually bind to it and block translation. And what do you think what's going to happen in block translation in a bacterial cell? It's going to die. It can't live anymore. Okay. So ribosome is really important. Okay. And specifically about the ribosome, the small subunits, and then you have the large subunit which has the binding sites for the tRNA. So the small subunits, and the mRNA is going to bind, and the large subunit is going to bind to that. The tRNA is going to bind to that to the mRNA, small subunit, and the large subunit is going to bind. And you have the A site, acyl site, where the acyl tRNA synthetase, making a protein. The P is the polypeptide site, essentially making elongating the change. And E is exit site. So this is where the this is where the protein is going to leave. So, okay. Actually, so newest studies being done shows that our RNA, including ribosomes, are actually really important to catalyze the activity. So it's not the proteins that are added to the ribosome, it's the ribosome and the RNA itself that actually catalyze the reaction. So these things going on in science, so in case you guys decide to research into this. So building a polypeptide, what are the 3 phases of building a polypeptide? And show the answer, but anybody besides I get your name? Huh? Anybody else know what the 3 parts of translation are? Yeah, correct. Good. So divided into 3 stages. So it begins with initiation. So basically, the mRNA is brought to a small subunit. mRNA is brought to a small subunit. And what's the start codon, by the way? What is the start codon? AUG. So AUG always goes from methionine. So unique aspect of every polypeptide you get from methionine. So AUG binds, and you have an anticodon. tRNA recognizes this anticodon, so it's tRNA, binds to the mRNA, to AUG, and with that, it goes for methionine. Methionine starts off with the P site. And when this binds, GTP has to be hydrolyzed so the binding could actually occur. So that's initiation. And initiation factors will then, after the tRNA and mRNA are together, will actually bring the large subunits. So you have extra proteins that help in the initiation. And then you go through elongation. So you have your protein that bonds. You have your protein, or your tRNA that binds to the P sites, and it has a part of amino acid on there. And next comes in the tRNA that binds to the A site. And then so, in order to recognize the correct codon, if matching occurs, GTP has to be hydrolyzed as part of the regulation of this. So once the right codon is identified, GTP gets hydrolyzed and allows the covalent attachment, allows it to attach to the large subunits. And then polypeptide is able to is added. Amino acids added to the polypeptide chain. Sorry about that. So the amino acids added to the polypeptide chain from this P to A site. So basically, this is moving this way, and the ribosome is moving to the right. So it's kind of like sliding back and forth. And when this happens, the reaction is catalyzed to form a polypeptide. So right here you have one amino acid here, the other amino acid is here, and then as a condensation, a dehydration reaction occurs, and the polypeptide becomes longer, so now you have additional chain. And then this will move over, this amino acid on the tRNA will move over, and then this will occur again. And the process of moving over is called translocation. Okay, it's called translocation. So we talk about elongation, codon recognition, peptide bond formation. Okay, and for the P site, peptide bond, P for peptide bond, P sites. Translocation occurs, and that causes the hydrolyzation of GTP, so it can actually physically move over to the next site. And then the last site is termination. That occurs at the E site, exit site. So termination is triggered by the stop codons. What are the stop codons? Anybody? Don't worry about it. No question. UAA, UAG, UGA. You go away. You are away, away you go. That's how let's get rid of somebody. Let's get that amino acid out of there. Or get that tRNA out of there. So it's triggered by the stop codon. And stop codon doesn't actually have tRNA but additional amino acid on, so it's actually just a release factor. So stop codon binds brings a release factor that comes in and essentially signals to the tRNA to stop, to the ribosome to stop bringing amino acids and duplicating polypeptide. And so then the complex assembles, large subunit goes away, tRNA goes away, mRNA goes away, everything assembles, and you have polypeptide.